Nick is in Jay Kazikadathu's lab and also formerly co-supervised with Dana Devine, and he is going to tell you about an awesome blood bag innovation.
Thank you. Thank you, Ed. Set a timer here because otherwise I talk way too long. Okay, so I have the advantage of getting to speak after a whole bunch of people that showed some great platelet storage data, so I get to very briefly kind of introduce the larger topic here. This research is fundamentally all about platelets and their storage, and we're familiar, right? Platelets, little cells in the blood, primary role is controlling bleeding and healing wounds. What I want to impress upon everybody is the diversity of people who end up actually needing platelet transfusions, whether you are a soldier on the battlefield, a mother in childbirth, a chemo patient, all kinds of folks need platelet transfusions in their lives. And so maintaining a large supply of high-quality platelets is essential to any good healthcare system. And that's where the problem starts, because the cells are very challenging to store. So in circulation, the cells live about 10-ish days, but due to a combination of different factors, they lose their quality. And that limits their shelf life, their approved storage time, to 5 to 7 days. So it's 5 days in most countries, 7 in Canada. And the problems that limit this are twofold. First and foremost is, as was just commented on, the platelet storage lesion. Throughout storage, the cells lose their quality, they activate, they aggregate, they start to die, their glycosylation patterns change, their shape change, etc. This is not a major focus of the talk today, but it is important to understand the rationale for the project. What will be the focus today is that of bacterial contamination. The platelets are stored at room temperature in media that contains a bunch of glucose and other nutrients, so any bacteria that get in can proliferate. These two problems in tandem create quite a large overhead challenge in terms of platelet supply and maintaining high-quality cells, to the point where up to 30% of platelets in Canada and globally can end up in the garbage due to expiration. Now, this is the problem that we're trying to solve, and so the way that we're doing it is through a material science approach. Platelets are stored in bags made of polyvinyl chloride with plasticizer. PVC is hydrophobic, and this hydrophobicity is a problem. Firstly, it drives protein absorption and subsequent platelet activation and adhesion, supposedly. And in tandem, there is bacterial adhesion onto this hydrophobic surface and the biofilm formation. But this all circles back to this sort of hydrophobic surface. And so the thinking is, well, what if you developed a super hydrophilic coating, one that can prevent the protein absorption, the platelet adhesion, the activation, and in tandem prevent the biofilm formation via bacterial adhesion? And I won't be talking too much about the formation of this coating today. The time just simply doesn't permit it. But if you do want to know about that, I'm poster 15. You can come and talk to me after the fact. It is what it is. What I want to focus on is how we are optimizing these coatings to actually kill invasive bacteria, not just prevent their adhesion, but to actually kill them off. To do this, we are leveraging antimicrobial peptides, AMPs. To perhaps oversimplify a little bit, AMPs are a family of proteins present in most organisms in nature, and they kill bacteria by interrupting their membranes and inducing osmotic lysis. And so it's known that you can conjugate surfaces with AMPs to render them self-sterilizing, and so this was the idea. If we can develop a super hydrophilic coating that prevents the protein and the platelet binding and can also kill bacteria via contact, self-sterilizing device that can hopefully extend platelet shelf life. The major key here is there is a wide variety of different peptides. They vary in biocompatibility, efficacy against different bacteria, and so we want to figure out which ones actually work the best. To do that, we need to screen their structure and identify those with the best qualities. To do this, we developed a high-throughput screen, and this is attributed to Helen, who was a previous PhD student in the group, and this is how she was doing it. So the coating itself is formed from dopamine that's used as a surface binder. If you look at this little schematic, it's these gray lumps. This holds hydrophilic polymers in position. Onto these different polymers, in, for example, 96-well plates, you can conjugate different antimicrobial peptides, one in each well. What you're seeing here is a cellulose membrane. Each individual dot is a different peptide sequence. We can take the peptides off, put them in the wells, fill the wells with platelet concentrate inoculated with bacteria, and that way identify the peptides that work the best. There's a whole computational side to this to identify the best peptides that we have. I'm not really going to focus on it, but this is how these peptides are being identified. And I wanted to give an example of how we're actually applying that into translating it into platelet storage bags. So this is an example of what the outcome data actually looks like. So y-axis, you just have the amount of bacteria in the unit. You have uncoated wells that contain only coating with polymer, so there's no peptide. We then loaded different one peptide here in various concentrations, 0.1, 0.15, 0.25 megs per mil, and a second peptide, same story. And what you will notice is that the peptides can kill off the bacteria. This is 100 CFU per mil staph epidermidis in platelet concentrates. The peptides can kill off the bacteria within 24 hours. E6 at all concentrations tested, TET20 only at the highest. That's that idea of different peptides work to varying degrees. And so we saw this outcome, we thought, okay, that's fantastic. Can we translate it into a platelet bag, actually make like a self-sterilizing platelet unit? And so that's what we went on to do. Okay, this animation looks great, awesome. We have these little 5 mil bags that we've been developing in the lab, and we can deposit our coatings with these different polymers, or with the different peptides. Fill them with platelet concentrate. Some of them are inoculated, some of them are not. That way I can compare the effect of the peptides on the bacteria, as well as just the effect of the peptides on the platelets. And they'll be uncoated with polymer, one peptide, the other peptide. Those are my groups. And I can say excitingly that this does actually work. So if I can attract your attention to this panel on the left, in black, you'll have the uncoated, red will be polymer, green and blue are different with the peptides present. Within 24 hours, they kill off the bacteria, and that sterility is maintained throughout. In contrast, you can see the growth of the bacteria up to 7 days in storage. So not only is the bag able to self-sterilize, but fortunately, we see good biocompatibility of the peptides. So the bags that do not contain any kind of antimicrobial peptide, the baseline platelet activation and the max clot firmness are compromised. We see higher activation, lower clot formation, but those that did self-sterilize are much lower, and indeed comparable to those that were never inoculated in the first place. And there's no negative effects of having the peptides in the storage bags, at least that we have detected. And so this is to demonstrate that not only can the units self-sterilize, but they are biocompatible up to long-term storage. This is at day 7 of storage, I maybe should have mentioned. And that's currently where we're at, this kind of proof of concept of the self-sterilizing platelet bags. There is more stuff that we need to do, compare against other blood-relevant bacteria. There's some proteomic characterization we want to do, further screen peptides, all that good stuff. But this is really just proof of concept. And thank you to all of the funding sources, all of our collaborators, and especially to the donors.
I get there first? Thank you, Nicolas. That was wonderful. So you mentioned about the biocompatibility, but can you comment on, any concerns when these products might eventually be transfused into patients?
So in terms of leaching, is what you're describing. So at this very point in the research, we have not looked at the leaching into the platelet concentrates as it currently stands. We know from previous studies doing this in for example, like isoosmotic buffers, that there is no leaching up to 21 days. And in the future, like in the distant future, when these things are finalized, these would more likely be peptide mimetics, so polymer-based rather than just peptides. So the ease with which they can be released from the surface, that goes down. They would be much more resilient in a finalized build. It's just the peptides are so convenient because they're these little building blocks. You can just change individual ones and screen an enormous amount. Doing this for 20, I think one student is going to check 20,000 peptides. So 20,000 different chemistries is too much. Okay. The peptides are the advantage.
Okay, awesome. Thank you.
You've coated the bag. Does it need to be polyvinyl chloride anymore? Can you use something with a higher oxygen ability to transport, diffuse oxygen, like polyethylene?
So what's interesting, and maybe this would be more of a question for the poster, when we deposit the coatings, it renders many surfaces much more comparable. So I haven't tested in non-PVC bags, but we have tested against multiple different plasticizers, and those we see, it renders all of the surfaces equivalent. Maybe polyethylene works. That's a good idea. I have not tried that. I'll definitely write it down. It would be worth playing around with, for sure.
Yeah, great talk. I was just wondering how much we know about which bacteria tend to predominate in this environment, or is it just a mixed bag of?
From looking at the stats, it's about 70% of it is staph epidermidis. The WHO has a list of bacteria that they find of high risk. MRSA is one of them. You have pyogeny, or Streptococcus pyogenes, Klebsiella, the name of the genus escapes me, but there are a couple, about 5 or 6, that are listed as high risk by the WHO. Those are the ones against which we would be testing this. But 70% staff.
Has anyone kind of systematically went through and cultured sort of?
Say that one more time, sorry?
So how consistent is it, bag to bag, these distribution of species?
So again, I think it's about 70% the Staph epidermidis. The other ones will make up the bulk of it.
Yeah.
Exact numbers I can't give you.
Thank you.
Sorry. Yeah, great talk. Maybe this is a naive question, but are there sort of risks for like resistance buildup to the peptides from the bacteria itself and biofilm? And have you looked at biofilm forming?
So we don't look at the biofilm I didn't put the biofilm formation slides in this, but we we count the number of biofilms, the size of the biofilms, and we look at them under SEM as well. We also see prevention of the biofilm formation, at least to the extent that we've tested it. Remind me again the first half of your question. Oh, the resistance. Just as bacteria can develop resistance to like a small molecule inhibitor, they can develop it to AMPs, although from what I've read, the degree of changes necessary for the bacteria to resist those AMPs is much greater. So it's not as easy for them to develop resistance. Certainly possible, but much, much more challenging. And that's part of the attraction for these guys, is that the biofilms are very antibiotic resistant. Having the peptides there is a much more effective strategy. Not only that, but obviously like they're on the surface where the biofilm wants to form, right? So it has multiple benefits.
Yes, so very interesting. So these peptides, do they come from Hancock? I don't know, but these small peptides obviously do wonderfully horrid things to nasty bacteria. Do they do anything to platelets?
In solution, I know that they are not the friendliest either. The extent of, it's much easier to see in red cells. The red cell hemolysis is much more evident. In platelets, the effects are much harder to detect. Over extended periods of time, you'll see the same patterns of platelet activation, shape change. You see more lysis of the platelets, for example. Platelet count starts to drop. The beauty of fixing them to the surface is that you lose a lot of that. The peptides are particularly effective in solution. Once you fix them in position, that activity and sort of cross activity goes down substantially.
Two questions. Have you looked at what happens to the dead bacteria? Do they stay in the bag as whole organisms, or are they degraded?
I actually have not. And part of the reason for that is because there's very few of them. Like if we load 100 CFU per mil and they're dead within 24 hours, that's going to be tricky to find. We could take a look on when you look at the SEM, you don't see any obvious bacteria still stuck. But that's after 7 days of storage, right, in the study that we have done thus far. What I do know is that if there are little bits of bacteria floating around, it doesn't seem to impact the platelets, but maybe that's a naive answer in that we haven't done all of the tests.
Second quick question, it's not a question, but it's to plant an idea for you in the future. There was a cluster of Acinetobacter septic transfusion reactions in the United States. A few patients were killed. And then when they started screening, they started finding Staphylococcus saprophyticus and Acinetobacter growing. So I'd be interested in you testing those.
Saprophyticus, we actually did something very, very similar for it. That's what Helen was actually working on in a different use case, more so for catheters. But that's interesting, and I'll definitely follow up on that. So thank you.
Very nice talk over here. Hi. Two questions. One is if the majority of the germs are staph epidermis-derived staph, is it just a handling issue? Because I mean, then this seems like an easy fix to me. The other thing is, are you concerned about when you kill the bacteria, because AMPs basically create holes in the bacteria? So are you concerned about toxins or whatever that then are secreted by the dying bacteria that could have negative effects?
So in terms of the sterilization, I'm assuming the overwhelming majority of contamination comes from the blood draw. So although we do a great job, I think it's about 1 in 1,500 units is contaminated. So that's very, very few of them percentage-wise, but it's also unavoidable that some bacteria may escape at some point. If there are ways of improving that, I definitely think everyone would be interested, but it's a challenge. Remind me again the second question that you had? Oh,
Yeah.
the fragments of bacteria, yes.
Yeah, or some toxins or whatever they are.
To the extent that we've tested, we haven't seen it. So after 7 days of storage, the fragments of bacteria are in there. It doesn't seem to impact the platelets. It's possible, and we have no transfusion model here. So, maybe once actually transfused into a mouse or something, maybe we'll see a difference. To the extent that we've done this, doesn't seem to be an effect. So hard to say at this point.